Hi, my name is Kelly, and I'm a fourth year chemistry major in the pressure lab at UCI. I started my research journey at the end of my second year. At that point, the only thing I knew was that I love chemistry, because it seems like a very hands-on and visual discipline. And research was a really cool way to explore chemistry outside of what I typically learn in the classroom or in the teaching labs at UCI. In the pressure lab, we developed non-invasive chemical tools to understand biological systems. Everything in the body happens at a microscopic level. Like you can't just look at a cell and with the naked eye and understand where all the compartments are moving, like what's interacting with what, and how a pathway takes place. But what I really love about my research is that it applies chemistry to these microscopic environments, giving us a way to actually visualize things that go on inside the cell. There's a really clever and impressive sort of problem solving involved with this type of science because you need to utilize all sorts of techniques to make the invisible visible, whether that's by using color changing assays or reactions that help you isolate specific biomolecules. My field of research is called chemical biology, and one interesting thing about this field is that there's always a balance to be struck between the chemistry and the biology. What I mean by that is you can't go full on chemistry because you're limited by the biological environment. You have to consider whether the reagents you're using are toxic to biological systems, if the temperature and pH are physiologically appropriate, and how the reagents will react in the biological environment around it. At the same time, you can't lean completely into the biology because a chemical understanding is necessary to manipulate the probes that you're using. The project that I'm going to present at the symposium, I think, does a really good job of striking the balance between chemistry and biology. Basically, what we're trying to do is incorporate a chemical structure into proteins as a way of labeling the protein. Rather than reinventing the wheel, we actually use the natural protein translation mechanism that all cells already do to build proteins. We're just using a mutant synthetase instead of the natural synthetase to incorporate our chemical structure into the protein. I've been working on this project for a little over a year, and it's definitely not easy. I've had to learn how to troubleshoot and revise my plans for the project whenever I hit a roadblock. This experience can be really frustrating, but at the end of the day, it's really rewarding. Our research is very important, even though it seems like such a niche field, because it helps us understand living systems better. And if you think about it, this research is complicated, but only because our bodies are. When I'm reading up on the literature in my field or I'm doing my own research, I'm reminded of how intricate and beautiful living systems are. And this just gives me a renewed sense of appreciation for the life all around me.